Happy to introduce our speaker today, coming to us from Colorado State University, Dr. Jessie Kremian. She's a research scientist working in aerosols and particularly in the regions of the Arctic and Antarctic, doing really cool stuff in the nexus between cloud physics, chemistry, and climate, as we'll see in this talk. So a little background on our speaker. Prior to working at CSU, she was at CU here in Boulder with Ceres. It's a NOAA scientist, I believe. Mm-hmm. And also did a postdoc at NOAA, but before that, got her PhD in chemistry from the University of California, San Diego in 2012, and a bachelor's degree in University of Illinois, Urbana-Champaign in chemistry. So yeah, works in aerosols and cloud and precipitation formation, and uses that information to understand downstream impacts on things like California water supply and clouds in the Arctic. And another interesting fun tidbit I saw in your personal bio was that you have this will apply to any of the older people, has a pet chinchilla named Biggie Smalls, which I thought was pretty cool. All right, so I'll let you take over, but I just want to mention for anyone that's online or here, Jessie collaborates with people here and will be visiting here again, living in the area. So if anyone would like to meet with her at any point, please get in touch with Paul Kuchera, Jessie, or myself, and we'll help set up some meetings because we'd love to foster collaborations with all the cool scientists in our area. So with that, I'll let you take over.
All right, thank you, Chris. All right. So I am basically kind of going to take you on a bit of a journey over the globe of various field studies that my team and I have done recently, looking at unraveling the sources of aerosols that seed cloud formation in the polar regions. So first, I'd like to acknowledge the students and the postdocs who are involved with most of the research on these different projects: Kevin Berry, Camille Mavis, Marina Nieto Caballero, and then Chelsea Beckmeyer. Okay, so the reason why we care about the Arctic is predominantly an effect called Arctic amplification, or polar amplification if you're dealing with the Antarctic. And basically what this means is as the world warms, these polar regions are warming even faster due to these feedback effects between the ocean and atmosphere, between the ice surfaces, so both glaciers and the sea ice, different types of cloud feedbacks, and then we have these intrusions of warm, moist air that are entering these regions. So they're actually warming much faster than the rest of the globe. And this is causing these frozen surfaces or these frozen landscapes to thaw even faster than they should be. So here's a plot of Arctic sea ice extent over the past several decades, and you can see that it's a pretty steady decline. When we look at the Antarctic sea ice extent, we kind of see that it hasn't really changed much in the past several decades, but then within the last several years, we're starting to see a decline in the sea ice extent surrounding Antarctica. Arctic permafrost, which is soil that's typically remained frozen for 2 or more years, that's in steady decline, so it's turning into more active layer soil. And then we have the Antarctic ice sheet, which is steadily declining over the past several decades as well. So all these different ice surfaces are melting, causing enhanced warming because you have more, less reflective surfaces that are being exposed more than their reflective counterparts. And so that's what's happening on the ground, but then clouds tend to play a pretty significant role in the surface energy budget in these changing polar climates. And so aerosols are very important because they help form clouds globally, but also in the polar regions. However, interactions between aerosols and clouds are particularly limited in the polar regions. And especially when we're talking about aerosols that come from sources like melting sea ice, from different land sources that are changing in a changing polar climate, and especially my area of research, which is ice nucleating particles, or INPs. And so there's a pretty dire need with these changing polar regions to be able to characterize INPs from these more regional polar sources and understand how they affect polar clouds in the changing Arctic climate and Antarctic climate. And so the reason why we care about INPs and clouds is because they can change the reflectivity of the clouds. So in theory, if you add some INPs to a cloud, you change the rate at which the cloud reflects solar radiation back to space. So you actually tend to cause a positive feedback where you entrain more heat into the Earth's atmosphere. Now, there are different ways in which aerosols behave as ice nucleating particles, or INPs. I won't go into all these different mechanisms in detail, but I'll focus on this one right here. And so this is called immersion freezing. And so it's basically where an aerosol first acts as a cloud condensation nucleus, or CCN. They form a cloud droplet, and then once that cloud droplet reaches sub-zero temperatures, depending on the type and the quantity of INPs that are there, it will actually form a cloud ice crystal. So basically, the INP is immersed in a cloud droplet first, and then it will freeze once it reaches the right temperature. And this tends to happen, this process is really important in mixed phase clouds. And mixed phase clouds, which contain both liquid and ice, are very prominent in the polar regions. So this is an example from a paper looking at the persistence of these mixed phase clouds that happen in the Arctic, which can last for days, sometimes up to weeks. So they're really important in terms of affecting the energy budget. So how we measure INPs are, at least what I'll talk about in this talk today, are using two different methods. So one being a cold plate and two being what's called an ice spectrometer. And basically what we do is we take samples from the field. So we can take aerosols that are collected on filters. We can take all types of other samples, so things that we think are sources of aerosols like seawater, freshwater, sea ice, snow, soil, plants. Pretty much anything in the real natural environment, we can mimic how a cloud forms or how they would form a cloud in a laboratory setting. So you can see by this video here, these techniques work very similarly in that we create an array of droplets on some sort of cold apparatus. We step down the temperature, and then you'll see the drops freeze depending on the types and the quantities of INPs that are present in those drops. And so from this information, we can calculate the INP concentrations in these different samples. And so I'll show quite a few of these spectra on the upper part of the slide here today. You could think of the bottom axis is cloud freezing temperature, and then the left axis is the INP concentration in these samples. And so these are cumulative over the temperature spectrum. And we know, not just by this analysis how many are there, but by the host of previous work in ice nucleation analysis, we kind of know what INPs are there based off where they fall on the spectrum. And so anything above about minus 38 Celsius, you need an INP in a cloud to initially form ice. You have to have them there. Things at the warmer, super cool temperatures, that's where INPs like bacteria and leaf litter are really effective at forming cloud ice. Now this middle regime here, it's pretty wide, but this is where things like dust, algae, fungi, and pollen can be ice nucleators. And then these colder temperatures, that's where things like volcanic ash and soot are really effective INPs. And so you can kind of see that these INPs come from a pretty small subset of aerosol sources, mostly natural. And so you can think of INPs as tracers for different types of primary biological particles, so microbes or proteins, different types of organics that come from microorganisms in soils, so those can be things like carbohydrates and other macromolecules, and then different types of dust, and these are the things that seed the cloud ice formation. Okay, so this is a map showing some of the more recent work that I've done in the Arctic. So I've kind of been all over the place on land and on ship. And so the first question I want to address for you is, what are the INPs coming from Arctic land? And so we'll focus in on a recent study in what's called Utqiagvik, Alaska. It's the northernmost point in the US, and it's in Arctic Alaska. And so there are different observatories that line the Arctic coastal region. So you can see Utqiagvik there in the red dot, but then there's other sites throughout the Arctic where there are different measurements of aerosols, meteorology, everything. Here is a plot from a review paper showing different measurements from some of these sites that are highlighted in the map of INP concentrations measured at these locations. So you can kind of see that even at one temperature, so say you look at minus 10, there can be several orders of magnitude difference in the concentrations of INPs, depending on the location and time of year. So one thing that really hasn't been evaluated in terms of INPs, or really aerosols in general, is permafrost. So you probably heard that permafrost is thawing with a warming climate. It's releasing reservoirs of greenhouse gases that have been stored there for thousands and thousands of years. But nobody's really looked at it from this perspective of, okay, well, what kind of particulate emissions might it be? putting into the atmosphere. So as I said before, permafrost is ground that's been continuously frozen for 2 or more years. It covers almost a quarter of the northern hemisphere. But with it thawing, it's causing all kinds of issues in these polar environments. And so you can see that it's causing road damage, infrastructure damage. It's causing coastlines to erode, which is a big problem for communities living in the Arctic. It's also causing ecosystems to change. So we're starting to see more shrubs growing farther north, and it's changing these ecosystems because of the types of greenery that are being introduced. But excuse me, could this thawing permafrost emit not just gases, but particles in the air, and could it actually affect clouds? And so this is basically a schematic of a hypothesis for a study that we did in Alaska. And so this is looking at kind of your typical permafrost landscape. You have the permafrost table, this layer that's thawing with climate change, and then the active layer soil, which is soil that goes through the annual freeze-thaw cycle. And what we think is happening is that these biological and soil materials that are normally preserved frozen in the permafrost are making their way into different types of lakes and rivers and streams in these Arctic permafrost environments, and then potentially making their way to the coastlines where they could introduce a lot of particulate materials into the ocean. And so once these particulate materials are in these water bodies, it's pretty easy for them to get kicked up into the air if you have enough winds. So you get bubble generation and you get waves, and those things can emit all kinds of particles into the atmosphere. However, these emission mechanisms for permafrost materials into the atmosphere are completely unknown. So we're really trying to test the initial origins of, does this make sense with how we think sea spray is generated, or is there some other way they get into the atmosphere? And so kind of a serendipitous study, after an icebreaker expedition I went on, I stopped at Fairbanks. And for those of you, who's been to Fairbanks here? Nobody? Oh. There's a really cool research facility. It's a permafrost research tunnel, and you can actually go in as a member of the public and just tour this tunnel that goes into like tens of thousands of years old of permafrost. But you can also collect samples. And so what I did is I stopped there and collected a few samples from the walls of this tunnel. And they ranged in various ages, so they've done a lot of carbon dating there, so you kind of know how old the soil is. And so that table is basically showing the different samples that we collected, including a number of different samples within the depths of a tunnel, but then also active layer soil on top of the tunnel, and also just within the permafrost table. So we had soil ranging from about 500 years old to all the way 30,000 years old. And so, first we want to know, okay, are these materials, do they have INPs in them? Because we don't care if they get in the air if they're not there in the soil. And so what we found was they were actually very rich in these ice nucleating particles. So here are those spectra, again, looking at soil samples. The different colors represent the different ages of the soil that we collected. And just to give you some perspective, these purple and pink spectra are now showing mid-latitude soils, so soils that were collected in the middle of the US, and then some glacial dust soil from Svalbard. And you can see that they look pretty similar. These pink and purple spectra have higher concentrations above about -8 Celsius. But then as you get to the colder temperatures, and really not that cold relative to cloud temperatures, these, especially the dark blue spectrum, really starts to kick off. So that oldest permafrost sample especially had more INPs than these soil samples that are studied way more often than permafrost soil. So that was kind of a surprising finding for us. So there must be something with this permafrost soil that's really rich in biological matter or something. And so that's why we went back to the field. So that was actually just, collecting a few samples in Fairbanks. We had a whole expedition team go up to the northern part of Alaska, and this study is called ArcSpin, the Arctic Study of Permafrost Ice Nucleation. So here's that map again, and now we're going to northern Alaska, where it's pretty much all permafrost underneath the landscapes there. There's a zoom in of the region, and then where we actually sampled, or where all those blue markers are. And so we sampled things like permafrost, water, soil, vegetation, aerosols, ocean water, you name it, we pretty much sampled it. And so we have hundreds of samples from this study. And I wanted just to highlight, like, all these little dots on the map are these lakes that are called thermokarst lakes. So you think, it's tundra, it's barren, it's a bunch of vegetation and soil, but really there's a lot of lake coverage there. So that can help support our hypothesis of, well, maybe there is enough water body surfaces to get that stuff into the air from the permafrost soils itself. Okay, so doing work in this region is pretty adventurous. You can't just really walk out onto the tundra and collect your samples. There are polar bears you have to worry about. And also, it's pretty marshy. So you can see a picture down there of me getting the ATV caught in what looked like a little puddle, but it ended up being a very big puddle. So it's a pretty challenging environment to work in. It's pretty fun. But we basically would take an ATV caravan out almost every day, and we would collect our samples, or we would go out on a small boat onto the lagoons and even into the coastal ocean waters to collect some water and aerosol samples. We actually had a foldable kayak that we would take out onto some of these lakes to get undisturbed water samples and also lake sediment samples, because these lakes are pretty shallow. They're typically only a meter to a couple meters deep. And you encounter a lot of wildlife. So that top picture of the polar bear was the very first day we arrived. This was during the pandemic, and we had to go quarantine for a week. And lo and behold, we see a polar bear eating a walrus on the beach. So that was pretty cool. There was the one day that we actually had some water sampling done just off the coast, and there was a pod of about 40 bowhead whales that went by. So that was pretty cool. So yeah, pretty adventurous, pretty fun. And then the video you can see, that's how we actually take out the permafrost cores. So what looks like you're drilling into vegetation, there's actually a lot of ice under the surface. Okay, so I'll just show you a highlight of some of the results from this ArcSpin study. So this is a map showing areas where we collected different water samples. The size of the dots represents the INP concentrations per milliliter of seawater at -15 Celsius, and the color of the dot represents the type of water, whether it's a lake, lagoon, or seawater sample. And so what you'll see is a lot of big green dots. And basically what that's telling us is that these lakes had the highest INPs of any of the water sources. And that, based on our hypothesis, might make sense because they're surrounded by these soil samples. When you go out into the lagoon, there's some, not as big blue dots, but they're still there. And especially this lagoon is pretty shallow, and so there was still pretty high levels of INPs in this lagoon just off the coast. Now the seawater samples were pretty variable. They didn't have as high concentrations in the water as the other sample types, but when we did see choppy seas because of the weather conditions we experienced up there, you would see a lot of sediment being kicked up, and that's when we did see the high INP concentrations. We also went to an eroding bluff. So this is where an actively eroding permafrost cliff line was actually thawing and releasing liquid into the ocean water. And so we collected some of this stuff, and it looks like chocolate milk. It's pretty rich in soil materials, and it's being eroded into the ocean waters. And so those water samples had relatively higher INPs than the other ocean water samples. And so this study is starting to wrap up. We're starting to have some students and postdocs publish on this work. We're looking both at the INPs, but also the microbial community composition through DNA sequencing, because we want to say, okay, what kind of microbial communities are there when we're seeing elevated INPs? Do we see some link between that? So we're looking at that in these water and soil samples, but also the aerosol samples to see if we see any links. We have a few publications, one that's basically summarizing the survey from Kevin Berry, and then Marina Nieto Caballero has a paper under review looking at the DNA observations from this work. So we want to look at the aerosol, right? That's why we care. Are these things getting into air? Are they getting into the clouds? And so for a lot of these different lakes, we would be able to set up these air stations that were upwind and downwind of them. And so these air stations contained a couple different types of aerosol filter samplers and then a small MET station. And Paul, you might recognize that. That is the 3D pause station. So we would set up these stations for a few hours around these lakes to try to get some idea of, okay, if we're downwind of these lakes, do we see more of these INPs? And anytime we did do this, we did see more INPs downwind. So you'll see the purple spectra here, higher than the gray spectra. We did see more INPs, and I don't show it here, but more aerosols also downwind of these lakes. So they're definitely generating some INPs. Now, whether they're linked to the permafrost, that's still something we're looking into. But that's where the DNA work can come in. So I will not go into a lot of microbiology because I'm not an expert, and also I don't think anyone in here is a microbiologist. But what we are seeing is some links between what types of microbes are in the soil samples versus the water versus the air. And so there are some very common similarities between these microbial fingerprints in these different sample types. And so 20% of the microbial taxa, or the different types of microbes that were there, that were found in the air, were also shared with the permafrost. So we know stuff from the permafrost is getting into the air. 33% were shared with the water, so we know a lot of stuff in the water relative to the permafrost is getting into the air. And 4% with both. So that 4% means we know the things that are common between the soil, the water, and the air. And so it's not a high fraction, but it could be that there's some sort of selective emission process, and this is something we're going to look into in the future. But the important thing is we're still seeing that connection. So there is still stuff getting into the air via our proposed hypothesis. Okay, just some general highlights from that Arcspin study. But what are the INPs coming from the Arctic Ocean? And so I'll just highlight some quick results from some field work in the Bering and Chukchi regions, so kind of off the coast of Alaska now. There's been a number of previous studies that have looked at INPs in marine environments. There's some nice studies that summarize these observations, including some of mine. Those spectra are showing kind of the range of INPs that have been measured off ships in the northern polar region. So you see there's pretty, again, quite a range depending on location and time. So I'll tell you about an interesting case that we saw during these Bering and Chukchi studies. So we luckily sailed at one point when there's a very strong phytoplankton bloom. So just to give you kind of a background story of this, a typical open ocean marine boundary layer does not have that great of INPs in it. Sea spray is okay. But what we saw was something a bit different. So this is kind of unexpected. So this map here is showing the ship track, and it's colored by chlorophyll concentration. And chlorophyll is basically an indicator for biological productivity in the water. And so you can see in the Bering Strait here, we have high concentrations of chlorophyll during both times when we sailed through. So we know there was a phytoplankton bloom happening there during this expedition. These dots represent places where, those are what phytoplankton look like, just in case you're aware. All kinds of weird little shaped critters. But zooming in on this particular region, because we know biological stuff tends to be good INPs, we wanted to look at this a little more closely. So now the ship track here in this inset is colored by the date in which we collect an aerosol sample. So we'll collect aerosols for a 24-hour period to make sure we get enough stuff to be able to measure. And then the dots represent locations where seawater samples were collected. And so looking around this phytoplankton bloom, these are the INP spectra from the seawater samples. And so you see the same color spectra represent the dates in this inset here, and the gray ones are just the INP concentrations from all the seawater samples. And you can see that the yellow spectrum, so the spectrum from August 29th, was on the highest end of what we observed the entire study. And so we're like, okay, well maybe all of these spectra around this bloom are really important, but that actually wasn't the case. So you'll see the other colors are actually on the lowest end of what we observed the entire study, which was kind of surprising. The aerosols tell a similar story. So even though these are over a wider spatial region, you get a little more variety of sources, so they're not as nice and straight as the other spectra. But you'll see that the yellow one, again, stands out for having the highest INPs, especially at the warmest temperatures when we expect biological stuff to be important. Okay, so there is a paper behind this. You're welcome to read the details there. It's a lot of physical oceanography and biogeochemistry and biological stuff and aerosol stuff and some meteorology. But I will basically explain this story in a fun schematic. Okay, so here's the region of interest. Here's the Gulf of Anadyr, which is south of the region we were in, Bering Strait, South Chukchi Sea, depth of the ocean. So as we're farther south, this is where we get really cold, nutrient-rich waters coming from the Gulf of Anadyr. And this is feeding the phytoplankton in that Bering Strait region. And so the phytoplankton are quite happy because they're getting all the food they need. They're becoming prolific in that region. However, there was also a sharp layer of salinity, a peak decline, so basically like a boundary layer of the ocean. And what happens is that blocks up the surface waters from reaching the bottom. And in this region, it's pretty shallow. It can be like 50 to 100 meters deep. And so the bacteria and the microorganisms in the sediment or at the bottom of the ocean aren't very happy because they're not getting any food. But as the waters start to move northward, the land starts to expand out, the waters will slow down. It starts to become more nutrient depleted because the phytoplankton are eating it up. They're not getting a consistent source. And then the phytoplankton are actually dying off. And so this is also where that picnic line breaks up and where the phytoplankton detritus is actually falling to the bottom of the ocean. But this is where the bacteria are happy because they're actually feeding on that detritus and they're becoming more prolific. Also during this period, we observed a storm in this region. So it was actually churning up water from that bottom layer to the surface waters within 5 days or less. So these bacteria were actually making their way to the top waters. And we saw them in the air where they were effectively being ice nucleating particles. So whole story come together by a bunch of different measurements, but it was kind of a cool observation that these phytoplankton blooms could be potentially pretty important for introducing warm temperature INPs into the atmosphere. All right, but as I was talking about earlier, ice, big part of the polar regions. What about when the ocean has sea ice? So I'll talk a little bit about some recent expeditions. Who's heard of the Mosaic expedition? All right, we got some people. All right, cool. And then another one, Artemel, that just happened last year. So the unique thing with sea ice environments, we studied this phytoplankton bloom, but with a sea ice environment, you have different ecosystems. And so what you have here is both this marine algae and bacteria that we find elsewhere, but then we also have sea ice algae and bacteria that are kind of their own unique communities. And so these different communities can emit things into the air that can then seed clouds, and those clouds can then impact the radiative balance over both the reflective sea ice surface and the absorbing ocean surface. And so our big question is, do these changes in these microbial communities affect the clouds? And so for those of you who aren't aware of Mosaic, it was a year-long expedition in the Central Arctic where the German icebreaker Polarstern purposely froze itself into the pack ice north of Siberia and then kind of drifted along with the transpolar drift over the course of the year. There were hundreds and hundreds of measurements, hundreds of scientists, different countries involved. And basically we were measuring everything from the bottom of the ocean floor to the upper mixed layer of the ocean, through the sea ice and snow, and then all the way up into the stratosphere. So quite a comprehensive study. This is a picture of what kind of the ice camp looked like, where we had different cities or catchy names for where we did special measurements that were surrounding the Polarstern. And basically for the INP measurements, what we did was collect thousands and thousands of samples. So we had aerosol samples that were collected on the ship. I had a little package that would collect mobile aerosol. It had a mobile aerosol filter sampler, which we called C3PO. There were also sea ice cores that we took, snow samples, seawater samples, and then we also collected water from open leads, which are cracks that open up within the ice exposing water, and then melt ponds, which are these basically ponds that develop on top of the sea ice in the summer as the surface hits 0 degrees. All right, so just kind of an overview of what the INP spectra looked like over the year. These are just kind of separated by calendar season. You'll see that the fall, I should probably use my pointer. Maybe not. Okay, the fall purple spectra, pretty log linear. There wasn't really anything above -15, so they were not that interesting. Same thing with the winter, kind of low concentrations of lower temperature INPs there. As we started to get into the melt season, that's where we start to see INPs start to appear at these warmer temperatures, so things that we would expect to be biological. But in the summer, that's where things really changed. We saw a huge drastic increase in the INP concentrations at these warmer temperatures. And so I was really, really excited because this was the season where we expect a lot of melt. This is where we have more open water, where we have more biology happening in the waters. So are these INPs dependent on the melt that happens during the summer period? All right, so we looked into a bunch of supporting measurements to try to describe this story and why it might be. This top plot here is showing also the INP concentrations, but now over time, with the darker blue markers indicating colder temperature INPs, so things that would be like dust, and then the red and warmer color markers would be things that were more biological in nature. And so you can see that INPs were generally colder throughout the fall and winter, but then these warmer temperature INPs were only present during the summer melt season. We looked at the sea ice concentrations around us. So this blue shading here is showing the sea ice concentration within a 1 by 1 degree around Polarstern. You can see we're mostly within the pack ice. There were some dips here and there. And then the markers are showing the fraction of lead water, so open water cracks, and then also melt ponds that were present around the Polarstern. And you can clearly see that during the summer we had the largest amount of open water. That was near the ship. This bottom plot is showing chlorophyll concentrations in the seawaters, samples that we collected throughout the year. Not very high concentrations throughout most of the year, but then again, that summer period is where we saw the more biological productivity in the water. And so these warmer temperature INPs were observed during more open water and when we had more biological activity in the water. So we think, okay, well, maybe these INPs are from these melt sources, from this ice and snow that's melting over the summer. So we are looking into some more information on this. So Kevin has a paper that he's about to submit looking at what types of INPs are there and looking at the DNA makeup of these things. And so here's an example of INP spectra during that summer period. We had really high concentrations. And we can apply different treatments to these samples to give us some information on what types of INPs are there. So when we apply heat treatment, that basically denatures proteins in these samples. So we know that, okay, those INPs were proteinaceous, meaning they were biological. When we apply a peroxide treatment, if we see a drop in INP concentration, we know those were organic containing. So the peroxide will basically wipe out things from organics from biology. And then what we have left are inorganic INPs that are things like mineral dust. And so you can see that during the summer sample, we had a ton of biological INPs there. That's pretty much the case of what we saw for that summer period. And in the winter is when we saw more of the inorganic stuff present, which makes sense because that's when we get transport of terrestrial things from lower latitudes into the Arctic. More DNA stuff, but basically what this is showing is just the sources of different types of DNA over the course of the year. And you can see that during the summer months, that's when we saw more of this marine and freshwater influence. So we know that these microbes in the air were coming from local marine or potentially melt sources. So again, still a work in progress. But this kind of leads us to this, what I like to call a sleeping microbe theory. So in the sea ice, you have all these microorganisms that are living in these small channels, in these bubbles. They're kind of sleeping throughout the year. So they're preserved, they're frozen, similar to the permafrost. But as the summer appears, we have more sunlight that spawns productivity in the ice and in the water. And that also causes the surface to melt. So we get these ponds that are on the surface. What we think is happening is that these organisms that are frozen in the ice and potentially the snow are making their way into this water. They actually some of them like being in the water, and so they become more prolific. They're producing these materials, and then in the summer, when we have different types of, melt pond dynamics, you can have bubbles that are generated that eject particles up into the atmosphere. So this is what we think is happening. We do see evidence of this. So we also had some melt pond samples collected during Mosaic. This is showing the INP spectra of from those melt pond samples versus the bulk seawater. So in the water samples, you could clearly see that the melt pond samples had more INPs than the bulk ocean water. So we know there's more INPs in there. We know they're probably biological. We compared the aerosol samples near these melt pond sources. So that C3PO package we would put right next to a melt pond. So the air would basically bring in air from over that pond and compare them with the filters that we had on the ship, which was about 30 meters up and farther away. And you can see that the orange and blue spectra agree pretty well, meaning that what we're measuring near the melt ponds is probably representative of what we're seeing on the ship too. So we think it's not just right by these melt ponds that these things are being emitted, but they're potentially have a more regional impact. And that makes sense given, I don't know if any of you ever seen a satellite image of melt ponds on the sea ice, but they're just everywhere. They're kind of like those thermokarst lakes. There's just these really pretty blue ponds all over the sea ice. So we think they could be potentially pretty important. But does this happen every year? So this was just during Mosaic. And when does it actually start? Because Mosaic was during the pandemic, unfortunately, the polar student had to leave the Central Observatory to go get people in May, which is a very crucial period because that's when the melt onset starts. So basically, the surface will start to hover around 0°C, and that's when you start to get the formation of all these melt features. So we kind of missed that really crucial period. And that's where this other expedition came into place. So not the same year, it was a couple years after, but we really wanted to focus on that key transitional period during the May-June timeframe. And so basically we sailed in kind of a similar area to where the Mosaic ship was at that moment in time. So this is the ship track from the Odin now, so it's a Swedish icebreaker that we went out on. We left Svalbard, kind of hovered around the Fram Strait region, and you can see that happen over the course of several weeks. The dots here in the image are representing where we had ice station work. So the the Swedes gave me full access to their helicopter, which was great, and I would take groups of students out to go collect samples near these open water features and to collect ice cores and snow and everything. So we got to fly a lot with Sven. He was great. Taught a lot of students a lot of cool things on the ice. And basically, this is just kind of summarizing everything that we did during the expedition. So again, trying to really link these different aerosol samples collected at different heights, different locations to these potential source samples. So we had, again, aerosols on the ship. We had different types of aerosol measurements, more enhanced than Mosaic this time, where we could get some more information on what types of aerosols were present. We had a filter sampler on a helikite that was deployed from the back of the ship, so we could collect aerosol filters above the surface. We also had a filter on this thing called the helipod, so it's kind of cool. It's like this little pod that hovers below the helicopter, and they'll do transits to where you can collect all kinds of information over a larger spatial scale. Then we had ocean profiling samples, and then, of course, the ice station work. We were just there last year. We have students and technicians really working hard to analyze all these samples, because collecting these samples is pretty easy there, but then the real work is hours and hours they spend in the lab trying to analyze them. And so we're doing the INP measurements. We're measuring chlorophyll, turbidity, all types of different aerosol characteristics, and then also looking at, again, DNA information to give us those microbial communities. And so at these ice stations, this is a pretty crucial part of this work because we really wanted to address these local sources. And so we would take a helicopter with a bunch of equipment, and we would set up these air stations. And so this is kind of similar to Mosaic and to the ArcSpin study, where we have a little package that has an aerosol filter sampler and an aerosol particle counter in it. We had, again, 3D pause, so these little MET stations have been really handy. And so we would deploy these things down one of these open leads. And so by that picture, you can kind of see how big these leads can get. We also collected these source samples. So we would do some ice coring to collect samples, ocean profiling, so we would collect samples at different depths. We should also collect information on, ocean salinity and temperature, and all kinds of information to give us some more physical oceanographic information. We collected slush, so this is basically ice that was kind of chipping off naturally from the main pack ice flow. And you can see that it's actually brown here. So there was quite a bit of algae in some of the ice samples that we collected. We collected the surface microlayer. So basically, this is a really thin film of organic materials that covers every ocean surface. It's pretty tedious to collect. You have to dip this glass plate in, and the organic film will stick to it, and you have to squeegee it off in a bottle. Took those students many hours to do that. So thankfully, they have a lot of patience. And then we did some snow pit sampling. And so I don't have a lot of data to show from this study yet. We're still kind of working up the initial data, but I will show you that there was this transition that we observed during this period. And so this is just showing temperatures from different source samples. So basically the surface lead water samples that we collected, the top of the ice core and the bottom of the ice core at the different helicopter ice stations. And you can see that generally over time, towards the end of the expedition, things started to warm up, which makes sense. We're getting into the June timeframe. And that's because the air temperature really warmed up. There was a lot of fluctuation because we had some storms coming through, but compared to the beginning of the expedition, we were about minus 10 C. And the end of the expedition, we were nearing the melt transition. So we didn't quite capture it, unfortunately, but we did get pretty close to getting a lot warmer in these different surface samples and in the air. And we saw this affecting the biology in the water. And so these bars now are showing chlorophyll concentrations from various samples that we took at those ice stations, so the water and the slush. And you can see that the bars are bigger towards the end of the expedition, meaning there was more chlorophyll, meaning there was more biology happening in the water. So what was interesting about this expedition is the ice was very, very thick, so it was hard for us to get north, and also the melt onset was about 3 weeks late. So we wanted to be there to capture the initial formation of these melt ponds, but unfortunately, it was late. And we did have melt ponds on, I'm not joking, the last day we were there, I saw a melt pond. I was getting ready to fly near the melt pond, but it was foggy and there was polar bears, so we couldn't go out. There were 31 polar bears we saw on this expedition, so it was pretty impressive. But they will not let you go on the ice when there are polar bears present, makes sense. So not so sad we didn't, we saw a lot of polar bears, that was cool, but we didn't get to sample the melt ponds. But I will show you kind of the transition of what happened during this period visually. And so here's a video I'll show. It was basically I did these outreach videos where I would put a GoPro through the ice coring hole just so I would use for outreach later to show people what it looks like under the sea ice. But they actually ended up being very scientifically informative. So I'll just play this, if it plays. Oh no, we tested it and now it's not playing. There we go. All right, you don't have to hear me speaking.
On our ice core hole.
But you'll get
We'll
the gist.
see if we see anything or if I leave this in the bottom of the ocean. All right. See you all later.
And so that's what it typically looks like under the sea ice. So this was at the beginning of the expedition in May. You could see these really beautiful blues, really a lot of topography happening. We actually captured a brine rejection channel, so that string there is really dense salty water that gets expelled from the sea ice. You see a lot of little floaty kind of ice chunks that are coming from the ice. You see all this I know it's really cool. You see all these different features that you wouldn't expect if you're not used to. I mean, nobody really sees below the sea ice too often, but it's pretty cool. All right, so this is what it looked like that day, right? Really pretty blue, lots of light penetrating through the ice. By the end, this is what it looked like. So we couldn't see anything. We would just see this algae that was floating around everywhere. And so it was pretty impressive how over that few week time period, even though we didn't get to the melt season yet, there was still a lot of biology happening in the water. And so we're hoping to link that with our measurements that we're doing from these source samples in terms of the microbial makeup, the INPs, and also linking that to the aerosol potentially. All right, so just to summarize this Arctic work, I had marine and terrestrial in my title. I apologize in advance. I'm not going to be able to answer the question for you because we don't know yet. But both sources can be important for Arctic INPs, but it's really highly time and location contingent. So permafrost can be a rich source of biological INPs that come from land, and they can make their way into lakes, into coastal ocean water, and then into the air. And in the Arctic Ocean, especially in these pack ice regions where there's a lot more biological productivity, they can be rich in INPs, especially when you have these open water blooms near the sea ice, but then also there's a mix of marine and terrestrial sources even in the summertime. So disentangling what comes from the ocean and from the land is actually really a lot more challenging than one might think. All right, so I've showed these plots before. How do our measurements compare to these existing studies? So here's a couple studies. So this is that Arctic permafrost study, and then that's another study of mine that I did in northern Alaska during the spring. I didn't get time to talk about it, but just to show you kind of where these fall. This was done in Arctic spring, kind of falls in line with what's previously been observed. But then these permafrost ones, so this is just the aerosol, much higher than what has previously been observed in the Arctic. So that's really important, right? We're just kind of breaking into this potentially rich source of INPs that can be making their way into the air. So I will be doing a lot of work on this in the future because this was pretty enlightening. And then looking at these marine studies. So these are also aerosol samples from the three studies that I mentioned. You can see that these, one in the Bering and Chukchi and then the Mosaic study, kind of fall in line with what's previously been observed. But for whatever reason, this more recent one, where we saw a lot of the biology in the water, they were on the higher end of what's been observed in the northern polar latitudes. So we're still looking into why that might be, but these studies could, they're kind of breaking the mold of what's been observed there previously, perhaps due to the changing environment. Okay, last map, I promise. I don't have any results from these studies because they are ongoing right now, but we are doing work in the other pole of the world. So I had mentioned polar in my title. I don't have results to show you other than to say we are doing a bunch of INP measurements on the opposite side of the planet. And so we've already done measurements on a few different expeditions that have left Tasmania, going down all the way to the Antarctic continent and back. So these were two cruises that took place last year. We have measurements of our aerosol filter samplers for both INPs and DNA that are happening at Davis Station. So those started about a year ago, and they'll be going on for another year. We have a lot coming up. So these are just names of different research voyages that are happening in the next 1 to 3 years that we'll be participating in. Ships tracks to be determined, but mostly going to be kind of in the same general region. And then we are also starting measurements at Palmer Station on the Antarctic Peninsula next our fall, their spring. So I'll actually be going down there to set up our filters at Palmer Station, where there's a bunch of penguins instead of polar bears, which is kind of nice. So a lot of different measurements. You know, the Arctic is pretty limited in observations, but the Antarctic and Southern Ocean region is even more limited. There's literally only a handful of studies that have done INP measurements down there, and a lot of them come from our group. So we really have some gaps to fill in there in terms of the INP concentration. So I don't have anything to overlay here because we don't have data yet, but we're working on it. So we're hoping to kind of fill in this a little bit more as we go through. And so over the next 5 years, we plan to double, if not triple, the INP data in the Southern Ocean and Antarctic region over both the Antarctic continent and the ocean. And so with that, I'd like to acknowledge funding and logistics folks, and then also people who helped out with these expeditions, and then also thanks to all of you for listening, and I'd be happy to take any questions.
All right, thank you for that fascinating, amazing talk. Really fun, and it seems like a lot of very rewarding detective work goes into it. So I'll be happy to take questions. I'll bring this back.
Your plot of the INPs from the Fairbanks permafrost, it looked like the permafrost that was 20,000 year old was really depleted in INPs. Do you know what the cause of that is?
Let me go back to it. This one, right?
Yeah.
Yeah, we don't know, honestly. We unfortunately, you know, like I said, this was kind of like a side trip after an expedition, so I just took the samples for the INPs, so we don't have a lot of information on what else was in there. So we don't know. It could be, I mean, maybe there's not a linear correlation with more INPs the older the soil is. There must be something with perhaps the mineralogical composition, or maybe there was different types. I mean, I would guess the biology is not homogeneously spread across the soil too, so we actually don't know for this particular one. It's a good question.
Also, if there's any questions online, please feel free to enter those. Any questions in the room?
Thanks. Nice presentation. So I was wondering if you also sample the clouds, or you see the impact of these, the ice nuclei in the clouds, and there's a way to see when you have low ice nuclei and when you have high ice nuclei concentration, how does it affect the cloud?
Yeah. For some of these studies, we did not have that information, but for the Art of Melt study, for example, we did collect on this tethered balloon. And so we do have INP measurements at the surface and at different levels, up to the cloud and even into the cloud. We have one student who's looking at a case study from that using an LES model to see, you know, if they were coming from actual ground sources. She's still working on that, but balloon measurements have been challenging for INPs. You have to collect enough material to be able to measure them, and that's not always easy when you're on a balloon in the Arctic. So there's more work recently where we're developing smaller samplers that can collect at higher volumes, so we can have enough loading to actually look at that more often. But unfortunately for Mosaic, it was supposed to pan out, but didn't happen, so we have no INP measurements above the ground. Aircraft, you know, traditional crewed aircraft are definitely a way to get around that. So there's definitely a lot of campaigns like the more recent ARC-6 ones or Caesar, so those are different Arctic campaigns that have happened recently. We've had measurements on those aircraft to look at what's in the clouds. But yeah, it's not common we have both. I wish we did. But I think we're heading to that direction in the future where we have the capabilities to have these smaller instruments that we can deploy, you know, in these little packages on the ground, but also in balloons to try to link them a little better.
So I have a question regarding the 20 to 30,000 year old INPs, which is, I guess, at the end of the ice age. Do you have an idea where the sources of those INPs were from? Were they remote or local?
Don't know. And this is where working with geologists would be very important. Because I don't have a lot of information on the soil from those ages in that location. I know they did carbon dating, so they knew how old the soil was, but yeah, I don't know. This is where you could use some information on the DNA in those samples and the mineralogy. But even then, I don't know if it's as easy as, in an ice core, you could definitely capture soil layers and you can see that. Or like glaciers, you can see where there was deposited soil every year. I don't know about permafrost because there's already soil there, so it's hard. It's a good question.
Any other questions? I did have one other one related to that. So is there a changing amount of INPs being fluxed into the Arctic with climate change in recent years?
Potentially, because, and this was part of that Arctic campaign, was to study these warm air mass intrusions. So we're seeing more and more of those into the Arctic. It is bringing aerosol from lower latitudes. During Mosaic, we had a warm air mass intrusion that brought, we didn't measure INPs during it, unfortunately, but it brought enough aerosol up to the Arctic that was equivalent to the pollution of a European city. So it can bring a lot of material up there. And during the Arctic winter, you get a lot more routine mid-latitude transport just because of the polar dome location. And so there's a lot of stuff that does come from lower latitudes, and we don't quite know how that's changing over time. I don't know if we have the observations long enough to really determine that. And the Southern Ocean is a really challenging one because there's just so limited observations, and they don't get that kind of routine transport. They're just so much farther away from terrestrial sources. You have this big, vast ocean, and yeah. So I don't know.
Just to follow up, and you listed, you've collected all these great data sets, and you have a long list of more you're going to collect. Do you have a sense of how long it will take till you can answer that question? The rest of your career or beyond your career? I'm just curious.
Oh, my goodness. Wouldn't be just me. It needs to be, in our community in general, we're trying to push for more longer term observations because a lot of the ice nucleation work that hasn't been done is, maybe you get a year here and there, but most of them are on the order of months. There's more effort now. So I was talking to you earlier about my DOE contract where we have these longer term sites that we're establishing more routine INP measurements at. So we have, 3 years of data in Oklahoma, and we're trying to get more longer term measurements to actually look at these more climate relevant changes. And we're working with, there's a network in Europe called Actris. They also have kind of these baseline sites where they do different measurements, and they're trying to establish also INP measurements at these sites. So we can see how things are changing over time. Because a lot of this is, we think they might be changing because we have more open water. We might see more blooming happening in the Arctic, but it's not based on evidence because we just don't have it. So it is a good question. Even if we have 10 years of data, what can you say about that at one site? It's tricky.
Okay, so before we send off our speaker, I'd just like to reiterate that if any of you would like to ever meet more in depth with our speaker, feel free to get in touch with any of us, Paul, myself, or Jesse, and she'll be probably visiting NCAR again in the future. And just like to thank again our speaker for the very fascinating talk and for coming down here 1 week after hip surgery and presenting to us. So thank you very much.
Thank you.